So, good morning and good evening, everybody. It's 9 a.m. at the West Coast and 6 p.m. in Europe. It's a pleasure and fantastic honor to host our spectacular speaker of today, Pamela Bjorkman. And it's really super excited to have you and super excited that you joined us. And of course, I want to briefly introduce you, and I will start with a little bit highlighting your most important work, which I found super significant and was one of the papers which has really inspired me a lot during being a PhD. And I think, we're all reading lots of papers throughout our life, and in particular the PhD times. And it actually rarely happens that you still remember when you first read the paper. And yours was one of those when I still remember when you had it. And we all know that this was the paper which has resolved the crystal structure of HLA-A2. And you showed us how this works with where the peptides are bound before we knew that peptides are important, MHC are important, and T cell receptors are important, but you put it all together. And so I really think this is one of the top 10 papers in the field of T cell biology that was super instrumental. Of course, I'd like to introduce you a little more systematically. So you are right now the David Baltimore Professor of Biology and Biological Engineering, and you are American Institute Professor at Caltech. Few years before that, you received a bachelor degrees originally from received a bachelor's degree from the University of Oregon, and afterwards a PhD degree in biochemistry from Harvard University. There, as a graduate student, and I assume afterwards as a short postdoc, you made those discoveries that I've mentioned with Don Wiley together, and then you moved to California, and you were a postdoc with Mark Davis at Stanford, where you worked on T cell receptors. Afterwards, you became faculty, first assistant, associate to full professor, all at Caltech. There's a long list of achievements and awards you have received. I'm just going to highlight a few ones because otherwise our hour would probably almost be over if I would read through all of them. You're a member of the US National Academy of Sciences, the American Academy of Arts and Sciences, and of the American Philosophical Society. You received the William B. Coley Award for Distinguished Research in Fundamental Immunology, the Gardner Foundation International Award, the Paul Ehrlicher Ludwig Darmstädter Award, Max Planck Research Review Award, the L'Oreal UNESCO Woman in Science North American Laureate. That's what you received in 2006. Cepellini Award from the European Federation of Immunogenetics, and many others. I cut it now short here in the interest of time. I'd like to briefly highlight that you have always been excited, obsessed with structures and using structures to explain biology. Presently, you are working very actively, and that's, I think, what we're going to hear much more about now, is structural correlates of broad and potent antibody-mediated neutralization to HIV and SARS-CoV-2, and to use this technology to inform immunogen design. Related to this, you're using a lot of 3D imaging techniques such as electron tomography to investigate how HIV, SIV, or SARS-CoV-2 infections impact tissues and cells. And of course, we're very excited to have you here and hear more about this. And as we all know, we usually start our webinars with the question that we choose from a pool. And I think you choose the most exciting one, which is, and I'd like to ask this now, which is, what advice would you give to yourself these days if you were 25 to 35 years?
Okay, well, thank you very much for inviting me to give this talk, and it's a real honor and pleasure. What advice would I give myself when I was younger, which I probably wouldn't have, well, I think I would have taken it, but my advice would be to work on absolutely whatever interests you most, because if you try to plan ahead and decide what will get you a job or what will advance your career, or, hopefully not what will make you famous, but if you try to figure out what is going to be the next trend, it might not be what you're really interested in. And I think you should always work on what really interests you, because of course science experiments don't usually work out, right? So we all have many failed experiments. So I think to stay in it, you have to be very, very interested in what you're doing. So I would advise you to go with whatever captures your passion.
So that means more passion and less strategy, which I think is a very, very good and important advice. I'm fully agreeing with all this. Okay, with this now, we can start the webinar. So if you would please open your slides.
Okay, just 1 minute. Okay, so I'm actually not going to talk so much about HIV because we're still very interested in working on HIV, but it's just everything's a lot harder. So I'm going to talk about some immunological principles that we've learned along with many others during the SARS-CoV-2 pandemic. So I'm going to talk about our efforts to make a pan-sarbecovirus vaccine to protect against SARS-CoV-2 variants and also animal sarbecoviruses without updating. So sarbecovirus means SARS-like beta coronavirus. And so if you look at this, you can see that a lot of coronaviruses have emerged from animal reservoirs into humans. And I think we all know about the two epidemics before the SARS-CoV-2 pandemic, the original SARS in 2022 or so, then MERS in 2012, and then SARS-CoV-2, which caused the COVID-19 pandemic. And then besides that, there are 4 coronaviruses that cause common colds in humans. And so there's a lot of interplay between animals and humans that we're concerned about, because I don't think this is not the last coronavirus related pandemic I think that humans are going to experience based on the past 20 years. Okay, so just some basic things about coronaviruses and about all viruses is that pretty much what they need to do in their life cycle is enter a host cell, make copies of themselves, and then make more viruses that are released from the cell. And so the parts that are going to be important for what we're thinking about are the spike, which is like many fusion proteins in related enveloped viruses. Envelope just means it has a membrane around it that it acquires from the host cell during budding. So this is a trimer, the spike, and then that binds to the ACE2 receptor, which is a receptor that regulates blood pressure, and it's on epithelial cells in your lungs and other organs in your body. And so if your body or a vaccine induces neutralizing antibodies in response to the virus or a vaccine, these can prevent viruses from infecting cells so that they don't turn them into factories to make more viruses. So that's what we'd like to do during after an infection or a vaccine. And so one of the things that's really important to know about the coronavirus spike trimer is that it has this, what I think is a very odd property, where it has receptor binding domains, or RBDs, that are stable and must be stable for the viral life cycle in either an up position. So here there's a trimer, so it has 3 RBDs and one of them's in an up position, or in a down position. And so only when it's up in an up position can it bind to the ACE2 receptor. And so I'll come back to this later, but I think this property of coronavirus spike trimers, which is not shared with, say, HIV envelope trimers or flu influenza hemagglutinin trimer or respiratory syncytial virus trimer, Ebola, and so on, is that it must be stable in these 2 different positions, so it exposes antibody epitopes when it's up, and it has to be up in order to bind the RBD. Okay, so when the pandemic started, we were collaborating with Michelle Nussenswag, originally on HIV, antibodies from HIV-infected donors, and he started doing single B-cell cloning of COVID-19 convalescent donors in February and March of 2020. And so Christopher Barnes, who was then a postdoc in my lab, he's now an assistant professor at Stanford, he did a lot of cryo-EM structure to add to the literature. So there were a lot of cryo-EM structures at that time of SARS-CoV-2 spike trimer, and a number of them had neutralizing antibodies bound. So in these, this is just like I guess 9 of his many 20 or 30 or more structures that he did. And I'm just showing you this each time the The S1 subunit has the RBDs on it, and that's going to be in gray. The antibodies are various shade, different colors. And then the S2 subunit that's responsible for fusion of the viral envelope and the host envelope, meaning the membranes, that's going to be in dark gray. Okay, so really what I want you to see is various monoclonal antibodies here solved. These are just their variable domains now shown from structures with the antigen binding fragments bound. And so what you can see is there's a variety of different ways that these recognize the receptor binding domain, or the RBD. And the RBD is the site of most of the neutralizing antibodies against coronaviruses. And that's in general, not just SARS-CoV-2, but the rest of them as well. Okay, so basically we saw some of these antibodies bound to only down RBDs, some of them bound to up or down RBDs. And in the structures, we sometimes saw, 2 down RBDs, 1 up, or 1 down RBD, 2 up, or various different orientations. And then after Christopher did this, he solved a number of other structures. Those were isolated by Davide Robiani in Michel's lab, and then later Vinci Wang isolated single B cell sequences from single B cells for monoclonal antibodies that were induced after vaccination, and there were no basic differences. So they're all different from each other. But from these, we established some principles for antibody recognition of receptor binding domains. And this is from one of Christopher's papers in 2020. So we called 4 parts of the RBD, class 1, 2, 3, and 4. So these are these epitopes for antibodies. And we're showing you just representative variable. These are the antibodies, and we're showing only the variable domains, and we're showing representatives of each of these classes. And so they have different properties. So for example, class 1 and class 2 anti-RBD antibodies, they block the ACE2 binding footprint on the RBD. So it's very easy to understand how they neutralize. Class 3 and class 4 don't usually overlap at all with the ACE2 binding footprint, but they can also be neutralizing. And they had various other properties that we detailed, like do they bind to an up RBD, and some can bind to both up and down, some can only bind when they're up. So we had that classification, and we noticed that, of course, other people have different classifications out in the literature, but this is the one we use. And of course, this is an oversimplification. So there are things in the middle of these epitopes we defined. And one of them we're very interested in is what we call class 1/4, because it's in the middle of class 1 and class 4. So it doesn't usually physically overlap at all with the ACE2 binding footprint, but through sterics, it can block ACE2, but it also binds lower down on the RBD, which is a more conserved region. So we're really interested in raising conserved antibodies, because what we want are antibodies that would bind not just to SARS-CoV-2 and not just to its variants of concern, but also to potential to the RBDs of potential spillover viruses from families of sarbecoviruses. So this is what we're trying to raise is illustrated on this movie here. So what you're going to see is a gradient. We take all the sequences of sarbecovirus RBDs, so SARS-like beta coronaviruses RBDs, and we plot it in a gradient that falls from orange, which means variable, so lots of different amino acids at that position, versus conserved. And when we plot that on the RBD, you'll see a pattern that we keep finding when we look at, for example, the variants of concern as well. So those are the down RBDs. Now you're seeing a close-up. That RBD in orange is down. It's going to flip into the up position. And now we're going to put that gradient on. And you'll see that the top of the RBD that's actually the ACE2 binding region, the receptor binding motif, and that is more variable. And sort of at the bottom, you see more of this purple. And so we would like to raise antibodies such as this one here. This is from a human donor, but it's a class 1/4 antibody, and this is from another human donor in Davide Robiani's collection. So humans can raise these antibodies, but the immunodominant region is going to be the class 1/2 region which is unfortunately in the orange part or the variable part. So our goal is to advance the slide. My goal is to advance the slide. Okay, there. All right. So now we're just looking at the RBD from 180 degrees apart. So you can see the class 1 and class 2 epitopes, or we're outlining, an example epitope for each, and they're in this orange region. So down here is the class 4 epitope, which is quite conserved. Class 3 is sort of conserved, but not quite as much. So we'd really like to get the class 4 antibodies. They are known, but they tend not to be as neutralizing, perhaps because there's some steric issues in accessing that part when it's on the intact trimer here. But so we'd like to get these ones that reach more towards here and can block ACE2 binding. So that's the goal. Okay, so how are we going to raise these antibodies? So here is where we had this theory that we were trying to accomplish this for influenza virus vaccines and for HIV vaccines. And we've been working on this for a number of years. But the idea would be based on this theory here about what might be going on when you activate a B cell in a germinal center with an antigen. So we multimerize our antigens for vaccines on a nanoparticle, and I'll tell you a little bit more about that. But for the moment, this is just some kind of way of multimerizing antigens. And the antigens are symbolized by these circles here. And then the orange triangle represents a strain-specific distracting epitope that is immunodominant that we do not want to elicit. But what's going to happen if we make a so-called homotypic nanoparticle, in theory, is you're going to get strong cross-linking between adjacent B cell receptors on a B cell. And so these are membrane-bound immunoglobulins, and so you'll get cross-linking here, which will favor elicitation of strain-specific antibodies against these distracting epitopes. But what if instead of injecting that type of immunogen, we inject a Mosaic immunogen where, and I think this is important, the antigens are arranged randomly on the nanoparticles, so it's unlikely that any two neighbors will have the same color, let's call it, of strain-specific distracting epitopes here. So the B cell receptors, probably certainly fewer can bind bivalently, and you may have fewer orange receptors that are able to bind at all because they can't access an orange antigen here. Okay, so now instead of that, what we're hoping would happen is that you would preferentially activate these purple type of B cell receptors, which bind to the conserved region, which is a little bit less accessible in the case of the RBDs on an intact trimer. But since you can cross-link, the idea would be maybe these would be favored. So the theory here is that by making a Mosaic nanoparticle, we could perhaps favor we'd give a competitive advantage to these purple types of B cell receptors. So that's the idea. And I just want to emphasize back to this orange and purple diagram is that it was constructed with SARS-like beta coronavirus RBD sequences from potential spillover viruses from animals, but the SARS-CoV-2 variant substitutions map, you see the same orange and purple type of diagram here. Okay, so some of the details about our nanoparticles. We're using the SpyCatcher SpyTag system that was developed in Mark Howarth's lab when he was at Oxford. And so one of the things he has done with this is he has made a nanoparticle that has 60 subunits, and that's called the MI3 nanoparticle. It's based on a designed cage that was produced by David Baker and Neil King at University of Washington, but the Howarth lab modified it to put this SpyCatcher protein on it and in 60 positions, and anything that has a so-called SpyTag, which is a 13 to 17 residue sequence, will bind covalently to SpyCatcher. So what you get here now is a decorated particle. So you can really easily make homotypic nanoparticles, but you can just as easily, if you express enough of the different antigens and you they're spy tagged, you can make Mosaic nanoparticles. So to remind you, what we're doing is expressing the RBDs alone of these sarbecovirus RBDs. They have a C-terminal spy tag. That this is an independently folded part of the protein, so that's not a problem. And this is a model of what this Mosaic nanoparticle would look like that Cheng Fan in my lab made. So there's actually a structure of the MI3 nanoparticle cage, and the dark blue things are spy catcher, and the different RBDs are represented by different colors here, and they presumably are arranged randomly here. So there's 60 this is a Mosaic 8, so there are 60 divided by 8 of any single type of RBD. Okay, so at the beginning of the pandemic, we'd been working with spy catcher spy tag thing for HIV. And so Alex Cohn, who was then a graduate student in the lab, picked 8 different sarbecovirus RBDs to make a Mosaic 8. And at this, so some details here, we've now, at the beginning, it was the original Washington 1 strain of RBD, but we switched to SARS-2, the beta variant, when we were worried that we had to keep updating that. It turns out we don't, but anyway, the thing we're going to work with is called Mosaic 8B. And Alex picked these from a paper by Michael Letko, who was written before the pandemic. He was at Rocky Mountain Labs, then in Vincent Munster's lab, and they had a paper about potential spillovers from animals of SARS-like beta coronaviruses. So he picked these other 7 to put on the particle. And then importantly, he deliberately left SARS-1, the original SARS-CoV, he left that RBD off because we wanted to do experiments where, for example, we would inject these nanoparticles, we'd compare Mosaic and homotypic, we'd inject them into animals, and then we'd look for responses to matched RBDs. That means the RBDs represented on the nanoparticle, or mismatched. And we'd look for binding antibodies and neutralizing antibodies in the serum. And Alex deliberately left off clade 3 RBDs from clade 3 sarbecoviruses because those are related by less sequence identity. So the ones we attached were related by 63 to 90% sequence identity to each other, but he left some off to see how far we could get recognition of mismatches. And then, of course, we got all these different variants, and this is an incomplete list here, but we started looking at reactions to other mismatches. And so we're all the way up to, we keep trying to keep up with the latest variants, but we keep looking for serum binding and neutralization of latest variants of concern. Okay, so now I'm going to, without showing you the data from the first 2 papers that Alex and others published, I'm going to just summarize the result. So first, in 2021, he published a mouse study where we showed broader responses to mice that were injected with Mosaic 8 compared to homotypic. Okay, so then he went on in 2022, and I'll summarize that paper. He did a study with Neeltje van Dormelaan at Rocky Mountain National Labs that included a challenge. And in that study, we showed protection from, so non-human primates were immunized with Mosaic 8B, and we didn't have enough monkeys to compare with homotypic in that case. But what we were able to do was challenge those animals with both SARS-CoV-2 Delta after immunization or the original SARS after immunization, and the animals were protected. So that was against it, 2 mismatched strains, because neither the SARS-1 RBD or the SARS-CoV-2 Delta were on the particles. But then what Neeltje did was we could do a lot more comparisons in not in mice. And so this is, as I'll talk about later, a stringent model of protection, because these mice have human ACE2, they're transgenic for that by a ubiquitous K18 promoter. And so when they get infected with a coronavirus, they rapidly lose weight and they die or are euthanized. So this is a very stringent challenge model, whereas most humans and non-human primates don't get that sick when they fortunately for us, when they're infected with these coronaviruses. Okay, and then I'll also not go over, this is a paper by Cheng Fan, a structural biology postdoc in my lab who solved a lot more cryo-EM structures. We made monoclonal antibodies in mice that were in response to both Mosaic 8 and homotypic immunizations, and those antibodies, then she solved cryo-EM structures, and it was really satisfying that she got 4 out of 5 of these were against this class 1-4 epitope, and the 5th one was against a class 3 epitope. So we were getting antibodies that we wanted from the Mosaic, and the monoclonal antibodies from the homotypic were not so good. So that's in this paper here. But then I will talk about deep mutational scanning that we've done, because that segues into our more recent study. So this is a way of mapping epitopes. So let me just summarize first the very stringent. So this was Vincent Munster convinced us that we needed a really stringent animal model of protection for these studies. And the reason he said that is because you really want to tell if there's any advantage to using Mosaic 8B versus homotypic SARS-2 beta. So you, as you may have seen in papers, most or all experimental vaccines will protect against a SARS-CoV-2 challenge in animal models. So in these very stringent animal model here, where the mice lose weight and die, we were able, what Neeltje did was to immunize with Mosaic 8B or with homotypic, and then challenge the model, challenge the animals with either SARS-CoV-2, so that's matched, or with SARS-1. Okay, and then you look and see what happens to the animals. And this is all published in the paper, but the summary is the Mosaic 8 nanoparticles were protective against a challenge by a matched strain, so SARS-CoV-2 beta, or a mismatched strain, which was SARS-1. And the homotypic immunized animals, when they were challenged, they were protected against SARS-CoV-2 as expected, but they were not protected against SARS-1. So I think this has 2 important ramifications here. The first one is that I think it means you could use Mosaic 8 or 8B now to protect against SARS-CoV-2, even though it has 1/8 as many SARS-CoV-2 RBDs as the homotypic. So I think it could be used now as a booster for protection. But more importantly, perhaps for our purposes, is it protects against a challenge that the SARS-1 challenge, and that was representing, we don't know what's going to spill over in another pandemic. And so we use SARS-1 to represent something that might spill over in the future, because there were challenge stocks available of SARS-1. And the Mosaic 8B protected against that unknown thing, but the homotypic did not in this stringent model of challenge model. So then I summarized it. We're using SARS-1 to represent an as yet unidentified sarbecovirus that could spill over into humans to start another pandemic. Okay, so let me just go on. That was, I was talking about that part. And then I mentioned that Cheng Fan solved all these structures, and just to say this very quickly, because she had the structures, she could take her model of the MI3 spy catcher nanoparticle and model in intact IgGs to represent membrane-bound B cell receptors, to sort of ask, is the geometry that you'd expect on these nanoparticles, would it support this kind of cross-linking between adjacent RBDs that are being recognized by, here representing B cell receptors binding to those conserved regions? And the answer was yes, the geometry looked like it would work out. Okay, so now I want to talk more about what these data here, which are epitope mapping using a technique that was developed in Jesse Bloom's lab at the Fred Hutchinson called deep mutational scanning. he developed it for influenza virus, but also for SARS-CoV-2. Okay, so the point here is that we had analyzed individual monoclonal antibodies, but of course the immune response that is produced in all cases is always polyclonal, not monoclonal. And so to be able to evaluate serum antibodies that are polyclonal, Jesse Bloom developed a deep mutational scanning approach, and we worked together with him and Ali Greeny, who was then an MD PhD student in his lab. So what they had done was to take RBDs and do yeast display. And so they introduced mutations in every RBD residue. So there are about 200, and they changed it to all 20 different amino acids. So there's about 4,000 or so individual RBDs that are each represented by a yeast presenting that RBD. And I should say that a lot of them didn't fold properly. So they had a lot of data that's freely available on their servers that tell you which RBD mutations stop, prevent correct folding. They then scanned these libraries to ask which mutations prevent ACE2 binding. They got all that information. They used these to look at monoclonal antibodies that were out there, and they could map their epitopes very accurately. You could compare that with structures of those antibodies bound to the RBDs. So they did all that. Then they did polyclonal serum analyses, and they showed where the serum from vaccinees and people who'd been infected with SARS-CoV-2, what epitopes were recognized. And they were the immunodominant class 1 and class 2 epitopes. Okay, so that's what was being elicited by the vaccines people had gotten, and that's what was elicited mostly by an infection. Okay, so we did this experiment with them where we took mice and we primed and boosted them in the presence of an adjuvant with either Mosaic 8B or homotypic, and the homotypic had 60 copies of SARS-2 beta, and the Mosaic 8B had 8 different RBDs on it. Okay, so these are the data these are compiled data from an average of 6 mice here. So the thick line is the average of these 6 mice, and then individual lines are in the lighter things. So what you can see is, first look at the homotypic, the response, the epitope mapping for the homotypic SARS-CoV-2 beta immunized mice. And what you see is all these peaks here near a particular residue, 484, that is in the class 1 and 2 region. Well, it's in the class 2 region. It's right over here. And so most of the mutations that affected the binding of the serum antibodies were in the class 1 and class 2 epitope. And this was very clear and obvious from this response. And it kind of looked like what you'd see in vaccinated or infected people. But it shifted, which was really satisfying to us. It was shifted towards the class 3 and class 4 regions in the Mosaic 8B immunized mice. So we were seeing something different, and that was what we were trying to do. We were trying to elicit antibodies to this class 1/4 region, and through a lot of mapping and so on, that looks like what we were doing. So we were getting mostly class 4 and class 1/4 antibodies from those immunized animals, but for the homotypic, it was mostly class 1 and class 2. Okay, so it looks like we were really targeting these purple regions, which is what we wanted to do. And so then people would always ask me, well, if you target the conserved regions, won't you induce mutations in them? And here's going back to the beginning of the talk, why I think it might be possible to make a pan-sarbecovirus vaccine in the case, because of this, what I think is a very odd property, or a unusual property that these coronaviruses evolved, and that's the ability to have their RBDs in 2 positions, either down, where it has to be stable, or up. And the interesting thing is that the purple regions are down here, but if you ask, excuse me, if you ask, see, the RBD has to be stable in the down position. And so it contacts the rest of the spike trimer in that case. And if you plot on the surface of the RBD, the regions that are in contact with the rest of the spike trimer, that's shown here on the surface in blue. And now this is from 180-degree rotation here, so you can see it from both sides. And now you look at where the purple or more conserved regions are, and you can see that the blue maps quite nicely onto the more purple regions. So I think that you can certainly mutate the purple regions, but not as wildly as you can mutate the orange regions, which are not in contact with anything else. So for structural reasons, I think you have to keep the purple regions fairly conserved. So I don't think coronaviruses can escape as easily from antibodies targeting those purple regions. Okay, so now I'm going to go into something where we're really trying to look at real immunology here using using these potential vaccines as experimental tools. And also it's relevant to our plans to do a phase 1 clinical trial with Mosaic 8B, which is being funded by the Coalition for Epidemic Preparedness Initiative, or CEPI. So obviously, if people get a Mosaic 8B vaccine, they would not be immunologically naive because they're either infected or vaccinated or both. So the question we've been asked is, could this original antigenic sin, or some people call it immunological imprinting, could that affect the immune responses to Mosaic 8B in non-naive humans? Because all of our experiments were in naive animals, all of our initial experiments. So the first question is, what is original antigenic sin? It's also called immunological imprinting, as I said. So I like this definition, which was on the Rockefeller University website when Gabriel Victoria and Ariane Scheepers, a MD PhD student in his lab, published a paper on molecular fate mapping of serum antibody responses to repeat immunization because they were interested in assessing original antigenic sin. And so the definition on that site is that, or the description is that it was originally described in the 1950s by an American virologist who'd been the son of a Presbyterian minister. And it says that he chose decidedly religious terminology to capture his frustration with the immune system's tendency to imprint on the first antigen it encounters, even to its own detriment. And so there, in this case, they're referring to the immunological imprinting that affects our responses as humans to various influenza viruses that we encounter throughout our life. Okay, so the reason I'm showing this is I'm going to show you at the very end some molecular fate mapping of serum antibody responses that we did in collaboration with Ariane and Gabriel. And so what I want to say, and I won't go into the details because it's really a system that they developed, but it's in their paper and you can look at it. There are mice that allow you to, they have either a strep tag or a flag tag, and you can then separate out antibodies that, if you do a repeat immunization, that are de novo antibodies, so they're elicited new, or they're recall antibodies, so they're the ones that were from the original vaccination. Okay, so we have a bioarchive paper now where what we were looking at was, I'm kind of summarizing it here. And I won't go into a lot of detail here, but we did comparisons of Mosaic 8B with homotypic again. This is all in pre-vaccinated animals. So they'd been pre-vaccinated with COVID vaccines. And we compared that to mRNA LNPs like Moderna or Pfizer vaccines. And we compared it also to an admix, which is you take Mosaic 8B and you make homotypic nanoparticles and you mix them in the same molar ratio. So you have 8 homotypic nanoparticles representing the composition of Mosaic 8, and you do all those comparisons. So I won't have a lot of time to go into the details, but it's all in the bioarchive preprint. But the bottom line is this kind of a summary. We got in the pre-vaccinated animals, we got the broadest responses in general to Mosaic 8B, followed by admix 8B, followed by homotypic. And the comparisons to the mRNA LNPs is probably not really very fair because we don't know what dose you should use in comparison to a protein-based versus an mRNA-based. But anyway, okay, it's all in the paper. Okay, so now here's very quickly with Deb Fuller and Sandra Droz at the University of Washington. They had some non-human primates that had been vaccinated 4 times with various nucleic acid COVID-19 vaccinations, and they were living there, and they kindly then vaccinated them at what we'll call week 0, which was 74. So over a year later with the RBD nanoparticles, or else an mRNA vaccine. Okay, so that's that one. And then we had 2 mouse experiments. We injected mice twice with something equivalent to a Pfizer mRNA-based vaccine. And then we came in 171 days after the boost of the RNA. So 192 days after the initial experiment with an RBD nanoparticle or else mRNA, sometimes twice. And then the Jenner Institute donated the AstraZeneca vaccine, which is an adenovirus-based vaccine that encodes the spike protein. So they donated some of that to us. So 2 prime and a boost with ChAdOx and then RBD nanoparticles. So 163 days later. And then these are all the different things we looked at. And this is now in my lab, was done by Alex Cohen and Jennifer Keefe. Okay, so in the pre-vaccinated NHPs, I will say this, like the great news was Mosaic 8B retained its RBD recognition breadth in the pre-vaccinated animals. So that was a relief to us. And it elicited cross-reactive antibody responses, even though the animals had gotten 4 prior COVID-19 vaccinations, and Mosaic 8B consistently elicited a broader antibody response than the homotypic or the bivalent mRNA LNP. And I'll show you just a little bit of the data here. There's a massive amount of data in this paper, and it's kind of confusing, but this is for the NHPs, and I already showed you the immunization, the pre-vaccinations and then immunization schemes. And so these are the results of ELISAs against a bunch of different RBDs. They're all defined over here, but this is in the form of these spider or radar plots. So the one with that's out the furthest, you can look at a bunch of responses here, and the one that has the most area in this case is the blue one, which is the Mosaic 8B. So it's consistently higher than the homotypic, and then the RNA is here in black. And so that you can't see statistics here, but if you do pairwise, if you gather these all so that each dot represents the response to a particular RBD in the ELISA, and you plot all these, you can do pairwise comparisons and get statistics. So Mosaic 8B in the pairwise comparisons was statistically higher than the homotypic and higher than the mRNA. Okay, so that's binding. And then we did neutralizations. And so what you can see is by comparison at week 2, you could see consistently higher Mosaic 8B than homotypic, and it kind of catches up a little bit by week 22. And in the paper, we present all the data for all the intervening 2, 4, 6, 8, all the intervening or most of the intervening weeks in the paper. Okay, so then I wanted to talk about, just in summary, without showing a lot of the data, I wanted to talk about the pre-vaccinated mice. Both, they were pre-vaccinated with either Pfizer equivalent vaccines or with the AstraZeneca ChAdOx1 adenovirus-based vaccine. Because those are mice, we could add more cohorts in that case. And so we compared Mosaic 8B to this admix that I already talked about, to SARS-CoV-2 beta, and then compared that to mRNA. And then we also used deep mutational scanning to map polyclonal serum responses in the ChAdOx pre-vaccinated mice. Okay, so again, the Mosaic 8B was the highest or the broadest responses, which is all in the paper. But what I wanted to show you was the deep mutational scanning, which we thought was incredibly interesting, because what you're going to see is, so remember, these are pre-vaccinated animals in all cases, and then they either got Mosaic 8B, Admix 8B, or homotypic, or mRNA LNP. And so these are the deep mutational scanning data. And if you remember, homotypic is the Washington 1 strain, and it produces class 1 and class 2 responses over here in this region. So that looks like it did before. And then you can map out where things are being recognized on the RBD. But the really weird thing, I think, is that this, for Mosaic 8B and Admix 8B, the response by deep mutational scanning is like very muted. And so we call that like a relatively flat escape profile. So you don't see much, and that's in comparison to what happened in the naive animals, where we saw, you know, a lot of peaks here indicating that mutations at positions in class 3 and class 4 affected the ability of these antibodies to bind those RBDs. So what's going on here? So we did some control experiments here, which I think are really interesting. So the first control is we did deep mutational scanning, and now at this point, Alex Cohen, with the help of Ali Greeny, had set up deep mutational scanning in our lab, so he could do all kinds of experiments on his own. So he takes monoclonal antibodies that we know where they bind, because some of them Christopher Barnes had cryo-EM structures of, some of them have structures in the literature. So we have a representative class 1, class 2, class 3, and class 4 monoclonal antibody, and we also have a class 1/4. So we look at what those DMS profiles look like, and they make complete sense based on what we know from those structures. And then what he does is he mixes them. So he takes an equal molar mixture of class 1, 2, 3, 4, and class 1/4. And when he mixes them together, he gets this flat response. And if he mixes, then he does other things. If you mix 10 times more class 3 and 4 and 1/4, so the ones against the relatively conserved epitopes, you see a little bit of stuff going on here. And if you do the opposite, you mix 10 times more of the antibodies against the more variable regions, you see a little bit of stuff emerging here, but it's not as much as you would see if you didn't have this mixture. So we've called these polyclass antibodies. They're polyclass properties, meaning they recognize multiple classes of epitopes on the RBD. They're in a polyclonal antiserum, but some polyclonal antisera are not as polyclass as others. So we're working on defining and setting up a quantification of how polyclass a response is, which we think we can do with analyzing these profiles here. Okay, so now I want to quickly talk about the molecular fate mapping that I mentioned. So the summary of that, and these are the last couple of slides now, is that what Ariane Scheepers and Gabriel Victoria did is they took, the results were that they primed and boosted mice with a Washington 1 mRNA LNP. So that's like the original vaccines from Pfizer and Moderna. And B cells that were primed by that dominated the responses after the RBD nanoparticle immunizations. And both Mosaic 8B and homotypic could boost cross-reactive antibodies, but the only way you could get de novo antibodies was with Mosaic 8B boosting. And so this is a summary of their experiments. They have these very fancy mice here that if you prime them with an mRNA LNP vaccine, you add tamoxifen to induce the ability to flag tag or strep tag these antibodies, and then you inject with some kind of RBD nanoparticle. And so we're comparing, in this case, Mosaic 8B and homotypic. And the results summarized here, but in detail presented in the paper, was if the mouse was pre-vaccinated with COVID-19 vaccines, like with Washington 1 twice, and then you could isolate recall antibodies versus de novo antibodies. We also did deep mutational scanning of the recall versus de novo. And basically the recall responses were coming out against the conserved epitopes, but we also got de novo responses from the Mosaic 8B animals, which we did not get pretty much at all from the homotypic animals. So we think that this is really interesting from a fundamental point of view. So these were the conclusions here, but really I'll summarize here now again. The RBD nanoparticle immunizations mainly produce recall antibodies, but de novo responses were elicited by the antigenically distant antigens that were on the Mosaic 8B nanoparticle. And this is something Aryan had seen in his earlier studies, which involved influenza virus, different degrees of relationships of their hemagglutinin. Now, so here's something that we think is interesting. Since the RBDs in Mosaic 8B are both closely matched, like 91% sequence identity, and distantly matched, like 62%, let's say, to the Washington 1 RBD that's part of the spike protein in the pre-vaccinations. So the mRNA LNPs used something encoding Washington 1 spike. We can use original antigenic sin to our advantage to both boost recall responses, and we know they're recalled to the part we want from the deep mutational scanning, but we can also elicit de novo responses. But in the homotypic vaccines are only, the homotypic immunizations only elicited recall responses. So here's another potential advantage of the Mosaic 8B. And this whole thing is consistent with the idea that the immune system evolved to respond to new pathogens. Because if it was absolutely trapped by original antigenic sin and could never elicit new responses, this would be a problem for all of us. So I think this is very consistent with an evolution of a very well-functioning immune system. So the summary is, from basic biology, the results inform OAS mechanisms. And I won't go into this, but it's in the paper. We think if you remove the SARS-2 RBD from the Mosaic 8B, we made a Mosaic 7, you get even better results in these pre-vaccinated animals. But from a translational point of view, it supports using Mosaic 8B to protect COVID-19 vaccinated people or people who've been infected against as yet unknown SARS-CoV-2 variants and animal sarbecoviruses with human spillover potential. Okay, so very quickly, the structural studies, Christopher's now at Stanford, Cheng Fan has done a lot of cryo-EM that I didn't have time to talk about. I wasn't able to mention the work of most of these other people. The original antibodies were Davide Robiani and Vinci Wong in Michelle Nussenswag's lab. And heading up the CEPI clinical trial efforts is Jennifer Keefe, who's also doing basic research, and Alex Cohn has stayed on as a postdoc to carry on these Mosaic experiments. I didn't have a chance to talk about Magnus's work on an mRNA LNP approach. He's starting to be a professor at the Gladstone Institute at UCSF in about 2 months. And these are our collaborators for the CEPI project. And I want to mention Vincent and Neelcha at Rocky Mountain Labs, and especially Gabriel and Ariane Schiefers and Jesse Bloom, Ellie for all her help helping us set up the deep mutational scanning, and the rest of the people here. So thank you for your attention.
Well, thank you, Pamela, for this really exciting and super insightful talk and for sharing all these cool insights with us. Now, of course, I'd like to ask many questions in particular about original antigenic sin and your concept, but as usual, we are cutting our discussion here short, and I refer everybody, including myself, to address any question to you via Twitter or now X, and we have your credentials down here, so please do so now. And with this, I would like to say thank you very much again for joining us, and also for all the people who joined us live or watched the video later online. And with this, I wish everybody a nice evening or a very nice day. And we look forward to seeing you again next week when our speaker will be Gérard Herbal, Abel, sorry, from Paris. So thank you very much and see you soon.